It's my pleasure to introduce Franck, Franck actually, he did his PhD in Paris with the lab of Serge Charpak with intravital multi-photon microscopy of calcium signaling. Then he moved to the Riken Institute in Tokyo, before coming back to France again as a university lecturer in Marseille, and he's a team leader since 2014, where he studies live imaging of cell interactions in normal and diseased brain at the Institut of Neurosciences in Marseille. I think Franck is a member of EU consortiums as a work package leader. He also has a few interactions with the industry. He's a co-founder of the startup EMXAP with spectral computed tomography. And he obtained a grant of the Institut Curie for exploiting cars in MRI imaging with some of industrial partners. He's very interested in neuroinflammation, in particular the interactions between immune cells and neurons in disease, and the role of certain subpopulations in disease progression. And I believe today you talk us a little bit about the context of glioblastoma and inflammation. So the stage is yours, Franck. Looking forward to hearing your talk.
Okay, so thank you to you and to the all organizers for this kind of opportunity to present the work of my team in front of such a nice audience. Over the past years, indeed, my aim has been to promote multi-photon microscopy as a preclinical research technique. And today I will focus my presentation mainly on the studies that we did on glioblastoma, with the aim to highlight both the technical challenge that we overcame and also to push forward some results that are used today to nurture our current research on neuroinflammation in general. So at the beginning, our aims was indeed to understand the role of microenvironment on tumor progression, and this using orthotopic glioblastoma models. And in this context, we wanted to know what was the specificity of brain parenchyma to develop tumor, and in particular regarding the high blood supply, the blood-brain barrier, the fact that there was some resident innate immune cell in the brain to protect it, and also what are the specificity of the physico-chemical property of the brain that regulate the infiltrative behavior of cells. And all these questions aim at answering the last question: why are brain tumors particularly devastating and incurable? So to do so, and to this aim, we have first to try to set up a focal orthotopic glioblastoma model that would be suitable to do recurrent to photon imaging. And this in order to assess how the tumor growth is isotropic or not in the brain. So to do so, we used a model of a U87 human cell line that were transfected stably with GFP, and that we grew as a spheroid of calibrated size in order to implant them in the superficial layer of the cortex. So they were injected in the cortex of nude mice, 200 micron in order to be close to the surface to facilitate imaging. That was in any case eased by the glass window. So this is pretty standard, but still our model has some specificity, and the specificity is to put a Cefadex B at the top to clog the injection tract for the spheroid. And the idea for this is both to prevent the escape of immune cell, of tumor cells that have been put into the brain, and that would follow the injection tract to escape, but also because this bead of Cefadex is glued on the dura mater, the idea is to restore an immunoprivileged environment, just the same as would be in control condition. And this is important. Why? Because we have observed that in the case the Cefadex bead was correctly glued, I hope you see my pointer here, then the development of the tumor is in depth and below the dura mater, as you can see here from in vitro image that were sliced in the other direction. But in case there is a mistake in the gluing procedure and that there is some space for cells to escape and go on the dura mater, then they grow typically on top of the brain, and pretty much like what is the case in a culture dish. So with this model, then we have tried to put effort on the quantitative aspects of the technique, and we took then advantage of the glass window to reposition the animal very precisely under the microscope at every session, so that it was possible, after injecting a vascular dye, to recognize some vascular landmarks in the prep, and to be sure that we were always, positioned in the same way and studying the same region of interest, concentric region of interest, in which we were measuring the fluorescence, either on the red channel in the case in order to monitor the vascularization, or on the green channel to monitor the tumor cells. So after repeated imaging session, we could show that, including injection, repositioning, and everything, the fluorescence measurement that could be obtained in reproducible region of interest was very, very much conserved. And the fluorescence was also, for the green channel, was correlated to the cell density. So using this very quantitative technique, we then addressed the question of angiogenesis, especially because angiogenesis is thought to be part of is part of the tumor development process, but we have found in the brain that it might not be the main parameter regulating the growth of the tumor. Indeed, we could show in some tumor that we repeatedly imaged that they were highly vascularized at some point, but then 10 days later, when the concentration of cell had increased dramatically, there were no further increase of blood vessels, no more blood supply. This was confirmed by quantitative measurement, where we saw on several mice that the blood supply became stable over time, while the glioblastoma cell proliferation remained. And at all time, there was absolutely no correlation between the fluorescence on the green channel for cell density and on the red channel for the blood supply. These in vivo data were confirmed later by immunocytochemistry of Debardieux staining. And we could show that in the place where the maximum proliferation was seen, it was not the place where that were the most vascularized. Nevertheless, we have used anti-angiogenic treatments such as bevacizumab in order to block, to prevent blood vessel formation and to block the blood supply. And in this case, as expected, we had a reduction of the tumor growth. However, once more, we were never able to correlate the decrease of cell density compared to a non-treated mouse with regard to the blood supply difference that we could achieve between the two animals. Furthermore, we found that this effect on tumor cell proliferation that modulated the proliferation rate of tumor cell was very transient, and after 3 weeks of treatment, we always observed some escape. As you see here, the tumor is somehow contained for 2 weeks, and on the 3rd week, it starts to explode, and this irrespective of the fact that there were no more blood vessels. So it's not a rebound of angiogenic process that would explain the recurrence of the tumor proliferation. So this argued in favor of an effect of bevacizumab rather on the stroma than on the blood supply itself. And to check this, what we did was to pre-treat the animal 1 week before tumor implantation in order to prime the environment with anti-VEGF therapy. And we could show in this case that the vascularization was not changed by 1 week treatment, so that when we implanted the tumor, it was in the same type of environment and it was if we would not have primed the environment. But the difference in tumor proliferation that we obtained was the same as the one that we had with curative treatment with bevacizumab. So most likely the bevacizumab was able to prime something in the environment, in the stroma. As a second evidence that stroma properties is the most important, we then used the pharmacological drug to block the stromal-derived factor 1 receptor. And although this drug was only mildly impeding the vascularization, we had a decline of tumor proliferation that was even better than with bevacizumab. So altogether, these different results seem to indicate that blood supply, at least in the case of glioblastoma, is not the most important, most critical parameter to affect tumor cell proliferation, and probably that something has to be looked in the stroma, where both molecular cues or immune cell or physical constraints can affect things. But because VEGF and SDF1 alpha were found to be efficient, and because these molecules are known to play a role on immune cell, we started to look also at the behavior of some labeled macrophages that we obtained after intravascular injection of rhodamine dextran. After repeated imaging, we can indeed get some labeled macrophages, and we use these macrophages to evaluate the behavior of macrophages with regard to tumor. And as you see, they really attack tumor cell and try to interact with it. And in the best case, they can eat and really phagocyte pieces of tumor cells. So most likely, and at least from this result, we thought that the critical parameter was immune cells. And we decided to set up the methodology to follow these immune cells in vivo. So for this, we didn't want to go for a complex labeling and decided to go for genetic labeling using available fluorescent mice at the Jackson lab, but crossing them together in order to generate a triple fluorescent mice in which neurons, but also resident microglial cell and peripheral myeloid cells are labeled in different color, respectively blue, yellow, and green. And this is an interesting mouse because we are able in this case to visualize microglia that are the resident immune cell of the brain. That is an immunoprivileged organ protected by several layers of meninges and an efficient blood-brain barrier. And this microglia, as you know, can get activated upon danger signal and then phagocyte what is dangerous and eventually release some trophic factors to heal the surrounding. But if the danger signal is too strong, then microglia starts to release cytokines to permeabilize the blood-brain barrier and to call the peripheral immune cells in the parenchyma. So basically, having these mice help us to study both the resident immune cell that protect the brain, as well as the second stage defense mode by calling the peripheral cells. So having this mouse, we decided to set up then a glioblastoma model that would be red instead of green to be able to separate the different color and to have it syngenic in order not to have destructive response as it would be the case if you would use U87 cells. And using this glioblastoma, red glioblastoma model, then we were able to visualize by 2-photon microscopy a very strong accumulation of microglial cell inside the tumor cells. Of course, this accumulation of cell is based on migration of cells, and everything is not static as it could appear from a still image or from a still image or a projection, as I showed previously. But we were able to see that all the green and the yellow cells were both present in the red tumor and were really sensing the environment and eventually touching each other, interacting with each other at the scale of minutes. And this migration over minutes was responsible for an evolution of the distribution of cells over days. And as you see here at the border of the tumor, a certain number of cells were migrating close to the tumor or displacing themselves inside the tumor. You see that this one has disappeared here and it was not present here. So there is really something plastic with immune cells in the glioblastoma. Because of this, after 3 weeks of tumor progression, we were able to see completely different pattern of recruitment of immune cell inside the tumor. And basically, what was particularly obvious and unexpected was that the microglial cells that were very much present at the beginning, at the early stage of the tumor, were much less present and reduced in density at the later stage of the tumor. And interestingly enough, as we could separate the two channels, we could see that the green cells that were mainly on the periphery at the beginning began to infiltrate inside the tumor on the later stage. This was, of course, observed on several mice and at different time points, so that it was possible to draw some evolution of the density of EYFP cells or GFP cells. And as you see, there is a decline. The decline that I presented earlier on still image is indeed present, and with a minimum around day 26, between day 26 and day 29. And interestingly enough, the density of green cell was instead increased between day 26 and day 29. So it is as if between these two time points, there was an immune switch inside the composition of the cell inside the tumor were completely different. But interestingly enough, the immune switch occurred at the final stage of the disease, at the time when we started to have an uncontrolled growth that led to death. These two markers were found also in the tumor of about half of the patient biopsies that we analyzed, meaning that indeed the peripheral marker and the microglial marker are most likely able to stage the disease, and possibly by ratioing the density of the two cells, these two types of cells, it is possible to set up a prognosis on the evolution of the tumor evolution. To know more, beside these two markers that were finally given by the fluorescent reporter, we decided to investigate what was the real immune profile inside the tumor. And for this, we extracted and dissociated several tumors at different time points and extracted the cells that were inside, and using 19 markers, surface markers, we faxed the cell and identified 14 population from the adaptive immune cells to the innate immune cells. So using these different markers, what was very striking was that the first month of tumor development after implantation is completely governed and controlled by microglia, as you can see here, with density of cells that are several orders of magnitude, or at least up to 10 or 20 times more than the T cells that you can have in the tumor. Because you must pay attention here that there is a broken line in the graph. But after the immune switch, what we observed was that first there was a decline of the microglial contribution, but then there was a massive recruitment of all peripheral immune cells. So this switch from microglial immunity to peripheral immunity seems pretty inefficient, if not deleterious. So the question is, why do the tumor cell escape better peripheral immunity than resident microglia? One possibility indeed is that the peripheral immunity needs to be boosted by a checkpoint blocker inhibitor in order to get a very efficient response. But one question that we might want to answer is it possible to favor resident immunity over the peripheral one, trying to prolong the effect of microglia and to prevent this, this income of peripheral cells? Most of all, what we need to know is what we can learn from imaging, because these cells are fluorescent. So when we classify the cells according to the three main classes, microglia being the resident, myeloid cell being the peripheral, and other leukocytes that are not labeled in our cells, it appears indeed that at the very beginning of the tumor, microglia and the fluorescent, the EYFP positive microglia that appears here in orange is the main regulator, both in number and proportion of the immune response. At the time of the immune switch, as illustrated earlier, we have a massive recruitment of peripheral immune cells, and these are mainly green, as expected, but we also observed that there was a significant portion of double-labeled myeloid cells. And this was somehow intriguing to us, all the more it was in the myeloid compartment. So going back to the gating strategy and checking the density of fluorescent cell in each population, it appeared that the double-labeled cells were in monocyte-derived dendritic cell of phenotype P2 or P3 according to our nomenclature. The nomenclature that we use is basically based on the idea that all the cells derived from monocyte come from green monocytes that are immature, and they have the choice either to differentiate toward a monocyte-derived dendritic cell phenotype with antigen presenting capability, or toward a macrophage phenotype. And these cells and the double label cell, they count among the dendritic cells at different stage of maturation. And this is important because these cells that have antigen presentation capability are able to educate T cells, B cells, in any case, adaptive immunity. And literature has also reported that they were able to dampen the activation of microglia. So basically, this double label cell that we have found in our mouse model seems to be a switch regulating the balance between the resident and the peripheral immunity, and possibly the adaptive immunity. So at least based on what is reported on the literature with the dampening of microglial activation, this is something that we have observed. So there is, at the time when this cell, the monocyte-derived dendritic cell, are present, so on day 28, there is a coincident decline of microglial contribution. But the other thing that was surprising is that with time, if you try to see how a P1 the fate of a immature P1 monocyte, it seems that with time, after the immune switch, the P1 monocyte seems to differentiate preferentially toward the monocyte-derived dendritic phenotype rather than the macrophage phenotype. Altogether, this means that the presence of these cells that are double-labeled and that we can recognize would sign for the moment when phagocytosis would be dampened, would be minimal, leaving place for tumor cells to proliferate again and again. Supporting this argument, Souberan and collaborators have recently shown on the same model that if you apply bevacizumab, you are able to reproduce the reduction, the dampening of cell proliferation in the early phase of the disease. But even if you continue the treatment, you lose this dampening, and even you have an escape of the treatment. You have a tumor that grows faster than if you would not have treated earlier. But interesting enough, at least with regard to what I was trying to point previously, it seems that the density of double-label cells that can be found in this tumor when it is treated is far higher than the density of cells that can be found earlier in the tumor, but also in the non-treated tumor at the same stage. So altogether, it seems that we have in hands a readout of the time when the treatment will start to be inefficient. So at this stage, this is about what I want to say, given the time that is allocated. But then the take-home message that I would like to carry is that we have set up in Marseille a dedicated orthotopic glioblastoma model that conserves the immunoprivilege of the brain and allows to study the deregulation of the immune response in tumor. We have mice in order to study the the dynamic of the different populations that are recruited. We have evidence that the anti-tumor effect of bevacizumab acts mainly via the stromal regulation rather than a modification of the blood supply. And this probably because we are already in an organ that is highly vascularized and where oxygen is already pretty much spread throughout the organ. Microglia is the first line of defense against tumor proliferation. And it is only later that the peripheral immune system is called to help. But the fact that it comes will finally dampen the possibility of microglial cell, phagocytic cells, to compete and to fight against the tumor cells. The double label cells then would contribute to this immune switch and to the escape from a bevacizumab treatment. With this, I want to acknowledge the work of all the collaborators who contributed to the result I presented today, and they are here highlighted in yellow, and as well as the financing agency that support the global project on neuroinflammation. And with this, I thank you for your attention and remain available for questions.
Thank you, Frank, for actually staying on schedule. And it's very nice talk. Thank you very much. So we have two questions. One is about if you claim that the blood supply is not the main driver of the tumor growth.
Yes.
But David Crossi is asking, did you see or investigate other features of the vasculature, such as diameter branching, that could impact immune cell infiltration?
No, we didn't investigate the diameter. For us, the blood, the fluorescence that is included in the voxel that we are recording account somehow for the diameter and the blood volume that can really accumulate there. So basically, if the perfusion should be small, then we would have a smaller fluorescence, or an higher fluorescence. It would be dense, but we didn't investigate further. We just kept on the evaluation of the blood volume.
So another question by Daniela De Martino is she's asking about this immune switch that you described. Yes. Is it dependent on chemokines production by tumor cells? Oh.
So this is complex and currently under investigation. It seems that both the tumor cells and the recruited immune cells can release this factor. So the composition of the immune cell at a given time would most likely influence the evolution of the chemical environment and also what the tumor will finally release.
Okay, we have another question by Roberto Weingart. He's saying that could you see that the immune response is altered by the large number of cancer cells that you actually implant?
I'm not sure. Can you repeat this question, please?
So he's asking about could actually the immune response be altered by the fact that you're injecting already a well-formed tumor, and in relation to patients that the tumor growth might be slower? So you're starting at probably later stages as compared to a patient.
So yes, I mean, at least from what we have in hands, what we believe is that at the beginning of tumor initiation, in any case, you would have microglial activation, and basically indeed, we implant a large mass of tumor, but because whatever the mass whatever the number of cells during the first month, there is no difference in the immune recruitment. Basically, the first 24 days, I would say, are driven by microglia. So the idea is that even if you would have a very small number of cells at a specific location, they would start to activate microglia, proliferate, and their proliferation would be completely under the control of microglia. And eventually, because there really is some factors, they would change the phenotype of microglia and try to dampen its activity.
So we are very happy to have Giorgio Seano today. So he's the head of the Tumor Microenvironment Lab at Institut Curie in Orsay in Paris. And he has been working for many years on aspects that have been already discussed today, such as tumor vasculature, vessel cooption, but also cell migration and radio resistance. So he got trained in Italy and actually got very familiar with his work where he studied tumor angiogenesis and integrins and provided one of the first evidence of this subcellular structure, which is the endothelial podosome rosette that controls sprouting angiogenesis. And he also developed the first ex vivo human angiogenesis assay. He then moved to the lab of Rakesh Jain at Harvard, where he investigated both the physical forces that are involved in brain tumors and how they affect neurovascular function, as well as looking at vessel cooption in glioblastoma. And then he moved to France, where he got a position, and he set up his lab to focus on brain tumors, microenvironment, and radio resistance. And he's currently funded by an ERC starting grant, and we are really looking forward to listening to your talk, who is entitled Intravital and Preclinical Vasculature Imaging in the Brain. Thanks a lot, Giorgio.
Thank you, Jackie. Can you hear me? Okay, yes. I hope so. Okay. Thank you very much for giving me the possibility to present my data here. I'm very, very honored because I follow this this session, all the session actually, with very high interest, because the intravital microscopy is in many different type of tumor is a niche, but all together really bring novelty to the tumor biology. So thanks for the introduction, and I'm a junior PI. Today I'm going to show you more or less mainly the work that I've done in Rakesh Jain laboratory in Harvard Medical School, with some preliminary results, some unpublished data coming from my own laboratory now set up in Institut Curie in Orsay. So let me just say two words on the general idea of this talk of the next 30 minutes. Today I'm going to talk about two stories, mainly two stories, all about an explored territory that is actually the normal tissue that is around the brain tumor. We are following the same topic of glioblastoma, but I will talk also about brain metastasis very slightly, but I will also talk about that. So in a very general way, in a very microscopic way, we can say that brain tumors in general are very, very deadly, both primary and secondary brain tumors. So both glioblastoma, for example, as Franck introduced, but also metastases that are coming from lung, liver, or melanoma. In both cases are very, very deadly. And macroscopically, we can define these two completely different types of tumors by saying that we may have tumors that are infiltrative or tumors that are nodular. So today I will show you one story that is talking about infiltrative tumors and is specifically on glioblastoma. We'll show you how tumor cells jump from a vessel to another one to really spread in the surrounding tissue. And in the second story that is more focused on nodular tumor, that are both glioblastoma and metastasis. I will show you how these tumors that create a mass effect that really deform the surrounding tissue in the brain and really damage the surrounding tissue with the force. So I will focus my interest in the physical force, in the solid stress that these tumors apply to the surrounding tissue, so damaging the surrounding tissue, killing the neurons, and reducing perfusion. So let's start with the first story, with the first project. So I am, as a background, I am a vascular biologist, and we studied for years and years how tumor actually call new vessel to form a new vasculature. As Franck was mentioning about bevacizumab, for example, was the top leading drug to stop angiogenesis. So angiogenesis is the process of formation of new vessels from persisting ones. This happens in many tumors, but I would say that there is a mechanism that is even more interesting because at least in brain or in organs in which we have highly vascularized networks, in which actually happen that the tumor that happen already to be quite close to blood vessel, already well perfused blood vessel, these tumor cells actually, instead of calling for new vessel, they actually follow the vessel. And so this phenomenon is called vessel cooption. And I want to underline very well the definition, because I will use the definition to really go through all of our results. So vessel cooption is the movement, and so it's an active movement of tumor cells towards and then along the persisting vasculature. So we are not calling about new vasculature, we are calling about the vasculature was a pre-existent in the surrounding tissue, in this case of the brain. And I say of the brain because obviously I'm focused on glioblastoma, but also because the brain is very highly vascularized and very well perfused. And so automatically the tumor cells happen to be quite close to something that is already a very good supply of oxygen and nutrients. So let's talk about very quickly about different strategies of infiltration in glioblastoma. Infiltration in glioblastoma is very crucial hallmark for the survival of patients. So glioblastoma can spread in the surrounding tissue with collective invasion, with perineural satellitosis, with diffuse infiltration, but also with vessel cooption. Pathologists call this vascular satellitosis. So they found, and this is also one of the hallmarks to define that this is a glioblastoma. They found many different highlights in the surrounding tissue close to the blood vessel. So we know that this is a case, this is happening in glioblastoma, but we have no idea how this is formed, how these satellitoses are formed. So we started the project with intravital microscopy. Of course, if I'm talking here, I will talk about intravital microscopy. And we massively try to understand how these satellitoses, vascular satellitoses, are formed by following with Fresnel window and high solution confocal multi-photon imaging. So when we implant patient-derived cell lines, GFP tagged in nude mice, in this case, this MGG8 is a mesenchymal cell type. This is a detail for glioblastoma. It's not so crucial for the general audience. And when we stain with label the blood flow with injection of dextran, big dextran, like 2 million Dalton dextran, we can actually see the 3D, with 3D resolution, we can see where the tumor cells go. And as soon as we inject in the brain, these cells love to stay close to the blood vessel. And so you see cells that are hugging the vessel, cells that are sitting, cells that are lying on the vessel, cells that are just touching with like a hand, cells that are really acquiring the phenotype that look like an astrocyte with the hand feet, or even cells that are capable to connect a blood vessel with another tumor cell. So someone may say, actually, my mentor, Jane, said, oh yeah, this is just random aggression. They happen to be close to the blood vessel, that's it. So we quantified in different regions, in different types of tumors, and at least in the surrounding tissue, in the infiltration of the surrounding tissue, in this specific tumor, I can tell you that more than 55% of the cells are actually close to the blood vessel. And so mathematically, I can tell you that this cannot be justified by just simple random aggression. So, okay, of course, I show you that they are close to the blood vessel, but I didn't show you that they are co-opting because, as I told you, co-option is a movement, an active movement of tumor cells towards the blood vessel. And so to do that, I need to do the dynamic imaging, and only intravital microscopy allowed me to do that. So we follow the same spot over and over again in the time, following the same region. That was not so easy, but it's definitely feasible with the tools that we have now. And you can see that after 2 days, 2 days after injection, we have this clump of cells in this representative image. Is we have this clump of cells that 3 days after go even closer to the blood vessel, and other 3 days after we go even closer. So with this type of imaging, we demonstrated that actually tumor cells actively go towards the blood vessel. Plus, we wanted to understand if on the top of moving towards the vasculature, we have also movement on the top of the vasculature, so along the persisting vasculature. So here you can see another solution intravital microscopy in which we do see one individual cell, always MGG8, that is moving on the top of blood vessel along almost 1 hour. And so you see, I mean, I am a cell biologist as a background also, and you can definitely see, I was really enjoying seeing this intravitally because you see all the steps of cell migration. And so you see the invadopodia or lamellipodia, difficult to say here in intravital microscopy, that is extended. Then these drag a new region that is farther, and then the whole body move. And so these loop is probably repeated over and over again to move along the vasculature. So, okay, I showed you that the vasculature, that these specific cells move on the top of blood vessel and they move towards the blood vessel. So I show you vessel cohesion. But glioblastoma is multiform, it's meaning that it is really, really heterogeneous. And so it's very naive to oversimplify by saying, oh, we demonstrated all of glioblastoma is actually vessel coating the vasculature. So to make a very long story short, we may now say that after a screening of different patient-derived cell lines, we may say that we can simplify with 2 different strategies of vessel coaptation. We may have cells that are doing individual cell vessel coaptation, or cells that are doing collective cell vessel coaptation. So you have example here. I mean, this is the same example that I showed you now. So MGG8, in which you see tumor cells individual. So tumor cells are really spreading in the surrounding tissue, and they are touching the blood vessel as individual entities. While we have other patient-derived cell lines in which you can definitely see that we have a completely different type of growth of the tumor, the growth strategy of the tumor, and plus you also see that we have a completely different strategy of vessel coaptation. So you see that you have this tongue of cells that really follow the blood vessel here, but also here. So it's mechanistically and in terms of cell biology, a completely different mechanism. So I mean, sorry, let me go back to this. Of course, trying to understand the difference between these 2 types of tumor is key, was key for our project. But I mean, combining these 2 is like comparing pears and apple, and so it's not really scientific, it's not really mechanistic. But I can tell you that these cells are highly expressed in OLIC2 and Wnt7. So we went to our collaborator at the time that was in UCSF, expert of OLIC2, David Rowich, and we asked him if he was able to provide us and to collaborate with us, obviously. This was a shared project with a mouse model in which we have the possibility to switch off and on OLIC2. And in this case, you can definitely see that tumor cells with OLIC2 are really, really spreading in the surrounding tissue, like individual cells. And you really see they are really around the blood vessel, like really individual cells. But when we take out OLIC2, you can definitely see that the growth strategy of the tumor completely changed. And you see that you have now a more bulky tumor with yes satellitosis, with yes co-option, but this is really like a collective cell vessel co-option. And the mechanism is completely different indeed, and I don't have the time to go through these, but trust me in this. Win7A and B are the actor of this OLIG2 tumor and these individual cell vessel co-option. And so the mechanism that you have in these cells is completely different from the mechanism that you have in the tumor cells that don't have OLIG2. So, okay, for now is all good for basic science, but I wanted personally, because I'm driven to try to solve something of the problem of the patient. I wanted to try to stop this process. I wanted to really inhibit with some target, with some drug, this target. So as I told you, this individual cell vessel co-option is driven by Wnt, and specifically by Wnt7. So we don't have a drug that is specific only for Wnt7, but when I was in Boston, Novartis is there, and they have a large battery of drugs that are against Wnt. In this specific case, we pick this LGK974, that is a porcupine inhibitor. It's capable to block the secretion of the maturation and secretion of Wnt, all Wnt. And so when you actually go back to intravital microscopy, when you use again this MGG8 tumor that is highly infiltrative, high vessel co-opting, and we use LGK974, and together treatment and intravital microscopy, we follow and we quantify the number of cells that are close to the blood vessel, you can definitely see that gradually the treatment strongly reduce the number of cells that are close to the blood vessel. And of course, we need also histologies to confirm this. Then the next point was to say, okay, if we are able to stop vessel co-option, are we also able to improve a little bit the survival? Because for now, vessel co-option could be completely independent on the survival and on the tumor growth. So in this case, we implanted again these MGG8 cells in mouse, in new mice, so immunocompromised, because we are talking about patient-derived cells. But we injected very, very few cells in order to allow the cells to really use the surrounding tissue, to really use the microenvironment to spread and to slowly grow. Obviously, this is this is expensive, time-consuming, but this was very important. So actually, from the moment of injection, here you see 20 days, but it's actually much more. This is from the moment in which we started analyzing, but these tumors take 3 months, more or less. So when the tumor starts growing exponentially, we added chemotherapy. So in this case, it's TMZ, timolizide. This is the most important chemotherapy in glioblastoma. So as you can see, the tumor growth slow down, but they grow back, as happened in patients. So in this setting, we hope more translational model of GBM. We also had the LGK974. So alone, LGK974 is not capable to do much, but when combined with TMZ, it's actually capable to improve survival. So I have also to underline here that we start treating tumors that are very, very late. So it's something that in general, in preclinical models, you don't do because it's very, very difficult. So we wanted to really try to reproduce what happened in clinics. So in the interest of time, I need to conclude this, to show you also the other story. So just conclusion very quickly. GBM cells, I show you that GBM cells co-opt blood vessel. I show you that OLIG2 and Win7 drive single cell vessel co-option, and that targeting Wins, in this case, all Wins with LGK974, is actually capable to improve chemotherapy mediated survival. I didn't show you anything about angiogenesis, but we finished this project also with an interesting consideration in which whenever we stop vessel co-option, tumor became more angiogenic, and whenever we stop angiogenesis, tumor actually became more vessel co-opting. So those are 2 alternative strategies. strategies of resistance mechanism. So in the second story, no, sorry, I forgot the preliminary result of the published data. So now what we are doing now, we are really interested in trying to understand the relevance, the real clinical relevance of vasculation. So this is something that really touched me a lot, because I don't want to do only basic science, but I want really to try to improve in some way in the future the therapy of these poor patients. And so we know that GBM cells can be intrinsically resistant to therapy, but what make the cells really resistant, and there are several proofs of that, are the niches. Could be the hypoxia niche, or could be a perivascular niche. So in the perivascular niche, we know that there is a microenvironment that actually allows the cells to really be protected, because it's done to protect also neurostem cells. So this microenvironment actually could be a shield for the therapies. So to really understand how this happens, we need to understand also how, which are the relationship with that, which is the relationship that these tumor cells have with the component of microenvironment of the perivascular niche. So I will show you just two other slides on this, and I will show you how we try to follow the relationship that the tumor cells have with the pericyte, actually in this specific case, more specifically with the smooth muscle cells. So to do that, we injected GL261, that is a mouse, is the model that actually Franck introduced before, this mouse model, and we injected in these mice that are alpha SMA this thread, and you can see, and we follow during the time, this spot of several different spots, but I will show you this representative image, in which you can see these tumor cells really spread slowly using the vasculature. In this case, this arterial with quite strong smooth muscle cells. So, okay, here you can definitely see that they use the vasculature, but the interesting part, in my opinion, was more trying to see really what are the relationship, the biological, the nano relationship of these cells. And so you see that really tumor cells touch the smooth muscle cells, but even more interestingly for me and for the literature in the last years, you definitely see some even nanotube that are maybe connecting with the smooth muscle cells, or even at the late stage, some smooth muscle cells that have been replaced by the tumor cells. So you can see here, but you can definitely see also in this case in which the tumor cells actually really start wrapping the arterial. Then the other component that is obviously important, and is important, has been showed also to be important for a resistant mechanism for endocrine factors, is endothelial cells. So for sure also we try to analyze the relationship of tumor cells, in this case red, with endothelial cells. In this case the endothelial cells are coming from are labeled in this RAG -1 type 2 GFP mouse. And you can definitely see also in this case that there are several situations in which really endothelial cells and tumor cells interact with each other. You see also here, and you can definitely see in different conditions in which really the interaction, the relationship between these two types of cells could be very important. What we are doing now is definitely try to reproduce this in vitro, and try to obviously go more in detail of the molecular mechanism underlying. So the second story, and I have not much more time. I have 10 minutes, even a little bit less, but I promise that I will stay in time, is on physical forces. So this project is always on brain tumor, but is focused on nodular tumor, as I told you. And so when I started working on brain tumors, was not specialized in brain tumor before, I really asked myself, what are the symptoms that really are important in the patients? and why the patients die at the end, not only for the tumor, but for the consequence of the tumor in the brain. So for sure, when you talk with a neuro-oncologist, they will tell you that everything is linked with brain damage. So the tumor can damage the brain in two different ways. One is biologically, and the other one is physically, with physical forces. So I wanted to really try to understand how the physical forces from the tumor actually damage the surrounding tissue. So in the interest of time, I will show you only the first part. For the rest of the part, you can go to the published paper. But this is, in my opinion, the most interesting part for the intravital microscopy. So I will show you how we were actually able to do in situ measurement of solid stress, and so this solid component of the forces that there is in the tumor. How we demonstrated that mechanical stress is actually capable to reduce vessel perfusion, and now we saw also that intravitally that we have a neural loss and deformation of the nuclei in the neurons in the surrounding tissue. So as I showed you, as I told you, we may have nodular tumor, infiltrative tumor. So this is in clinic. These are GBM, glioblastoma, but these happen also in metastases from other type of tumors. So to really simplify, to really have the possibility to modelize these in vivo, in preclinical models, we use two different models. One is UAT7. It is very, very nodular. We are not interested in the biology of this, but on the physics of this. And we use NGG8 also, that you already saw is a very infiltrative tumor. So with this tumor, what are the forces that are inside the tumor and exerted to the surrounding tissue? So to do that, we cut the tumor in half. So let me just explain you what is solid stress with a very simple, probably oversimplified example. So let's say that we go on vacation. So we hope after this COVID period to be able to go on vacation. We put all of our dress in a bag, we sit down on the bag and we close the bag. When we do that, we are putting a lot of material, solid material, in a confined space. This is what happened in the tumor. So when you arrive to the hotel, you open the bag, what happened? Happened that the dresses bulge up, okay? So, and this is what happened also in the tumor. So when you cut the tumor in half, and this is ultrasonography image, in which you can see that the tumor actually bulge up and take more space. And so you are releasing the stress. So without going too much in detail, mathematically, you can push back this bulging and you can quantify how much force is in the surrounding tissue, exerted in the surrounding tissue. And as expected, the nodular tumor are actually capable to push the surrounding tissue more than the infiltrative. And this was something that you can hypothesize by thinking, but was never been demonstrated. So what these UAT7 tumor, or anyway nodular tumor, are capable to do when they really compress the surrounding tissue. So here you have optical frequency dynamic imaging, that is intravital tomography of perfused vessels, and you have GL261, that is another pretty nodular tumor. And you see the whole cranial window in which you have one hemisphere and the other hemisphere, and the sagittal sinus, and the same mouse before and after that the tumor grows. So you can definitely see that actually there are several things that happen with the tumor grow. You see that we have the sagittal sinus shift. You see that we have the diameter reduction of the sagittal sinus. And more importantly, you see that you have an overall reduction of the perfusion of the vessel. And so remember that this is perfused vessels. And so we are actually capable to see only the perfused vessels. So this reduction is actually quite important. And you can see here with other two examples. This UAT7, again, ultrasonography, and you see this vessel is out of the tumor is growing here. That is actually reducing in diameter, and you will see also here with the breast cancer tumor in the contralateral hemisphere, these blood vessels are actually reduced in diameter. And again, you see this with the multi-photon imaging, in which you can definitely see this blood vessel that has a specific diameter, that slowly, when the tumor grows and push the surrounding tissue, is actually this diameter reduced more and more. So it's really like compressing a tube. So if reduction perfusion is one of the consequences of physical forces, the second consequence is for sure compression of the neurons. So we did a lot of histology on this. We demonstrated many other ways. You see far from the tumor that the nuclei are very rounded and are very large, while close to the tumor, you definitely see that the tumor are squeezed. And so these, together with many other proofs, show that we actually have force that actually is close to the tumor. So in the last point that I will not show you, we also demonstrate this more mechanistically by applying the force or releasing the force, and we found a neuroprotectant. I will skip these because it's just a repetition of what I showed you before. Obviously, I want to acknowledge all big laboratory for Rakesh Jain when I was there, plus our collaborator in UCSF that helped us in working on vessel cooption, and our collaborator from Oslo that was helping us with a huge analysis on patient, because we confirm all of solid stress data also on patient in MRI. And obviously, I want to acknowledge also my current laboratory in Orsay, in Institut Curie, and thank you very much for your attention.
There's 3 questions already. The first one was from Alexandra Chikina, and she would like to know whether vessels actually call for tumor cells or attract tumor cells along them, or whether tumor cells follow already preformed gradients of chemokines to find the blood vessel.
Yeah, we have proofs now. We didn't show anything on these in that paper, but now we work, it's 4 years that we work almost only on that. Yes, they actively call for tumor cells. I mean, wait just, I hope, a small time, and we will be able to publish that. These, together with the fact that these chemokine are also important for the resistance mechanism. And so they really also help the tumor cell to resist to therapy.
Then we have 2 questions of Roberto. The first one is related to Wnt. I think it's the LGK inhibitor from Novartis, and he wants to know whether this inhibitor is still reducing the number of cells that associate with the blood vessels in presence of temolizumab.
I guess so, but we honestly never done this specific analysis, because to do that, we need to do specific intravital microscopy, at least for that type of quantification. So I would say yes, but I don't have the perfect proof of that.
Okay. Then his next question was actually something that I would also love to ask you maybe later, but whether the vasculature is somehow leakier at the interface between cancer cell and the vasculature at the spots of cooption, or does it affect any type of like hemodynamic properties when you have cooption at these sites?
Yeah. Okay, there is our own data that show that suggests that, let's say. Plus, there is also another paper showing this, a very good paper on Nature Communication 2 years before us, in which they actually show that the tumor cells actually are capable to take out the astrocyte and fit. And so in this case, automatically they will release the blood-brain barrier. But this, and this is instead our data, this is happening only when you see collective cell vessel cooption and not when you have individual cells. And so in terms of glioblastoma biology, this is very important, because it's meaning that in the infiltration, when you have individual cell vessel cooption, you don't have leakage, and automatically you also don't detect this tumor with MRI, because in general you detect the tumor with MRI with the consequence of the fact that that you have leakage in the tumor. And so probably we are missing, and this is one of the reasons that glioblastoma regrow back, because probably we are missing regions that are really with a lot of infiltrated tumor cells that we are not seeing by MRI.
Bojana, who is part of the organizing committee, so she needs a little bit less of an introduction, but still I will introduce her a little bit. So she graduated actually in chemistry from the University of Belgrade, where she worked on analytical chemistry, and she isolated active compounds from endemic plants, which I didn't know, but I learned that today. But then she took a big turn, as she mentions, when she for the first time saw a fluorescent microscope, and she ended up developing lots of technologies for automated 4D imaging and analysis at the first stage of in the stress of studying malaria division and metabolic processes. She did a postdoc, and this is where I get to know her at the Albert Einstein College of Medicine, where she developed new tools. So this was in the lab of John Candelis, including photoconversion, which is something that is very interesting in using mouse models, and she definitely contributed to the current view that we have on tumor cell intravasation mechanism in a realistic microenvironmental context. So now in her lab, they are investigating the mechanisms of tumor cell metastasis by implementing established and developing new microscopy techniques at multiple scales, combining this with approaches from cell biology, tissue engineering, and systems biology into a systems microscopy. So the focus right now, and I think it's mostly the focus of today's talk, is the analysis of the tumor microenvironment complexity, including extracellular matrix remodeling by tumor cells themselves, and probably through invadopodia, which I guess is the subject of today's talk, which is entitled, real-time imaging of invadopodia and the perivascular niche. So, Bojana, the floor is yours. Thanks a lot.
Thank you very much, Jackie, for a kind introduction. So let me share my screen. All right. So
Perfect.
I'm perfect. Yes. So I'm currently at Temple University College of Engineering in Philadelphia, and also in the Fox Chase Cancer Center, which is a couple of miles further north. And I'm going to tell you again about the perivascular niche, this time in the breast cancer. So our lab is focused on looking into different steps of the metastatic cascade in the breast carcinoma. And for those of you new to the breast model, it mostly starts in the ducts, and you can imagine that this is a cartoon of a cross-section of the mammary duct, where the transformed cells start growing uncontrollably. At the time, they're surrounded by the basement membrane, and then this is again, simplification. I'm missing myoepithelial cells and so on, and there's collagen fibers around them. And so what happens? So this is what we call the stage 0, or the ductal carcinoma in situ. And what happens next is something that we as a field are still trying to debate on. It's either that the proliferation of the cancer cells is pushing on the basement membrane and collagen mechanically, and then basement membrane bursts, or there's a collective invasion, or maybe there's active degradation of the basement membrane by invadopodia, and then invasion into the healthy tissue. So that question is still open, and that's something that we're working on. But this is what we call invasion step. After the invasion step and basically the healthy tissue being recruited into the primary tumor, we go through the angiogenic growth and so on. Collagen fibers become really elongated, and sometimes aligned, and this becomes convenient highway for the cancer cells to migrate on through the stroma. Finally, some of the cells are going to reach the vessels, and then they're going to intravasate again, passing through the basement membrane. Now, keep in mind, this basement membrane is slightly different in components, so we don't know that the biology of the crossing across this basement membrane is exactly the same as the first time. So this process, as I said, is called intravasation. Cancer cells end up in the blood vessels, and with the blood flow, they're taken to the secondary organs, which in case of breast would be mainly lung, but also liver and potentially brain and even bone. But we primarily focus on the lungs. So you'll notice all of these steps of the metastatic cascade involve and are driven by the cancer cell motility, which is kind of convenient for people who like doing real-time microscopy. And the approach we take, and this is something that is becoming more widely accepted approach, and that's the combination of fluorescent dyes as well as fluorescent proteins, working 2D and 3D cell cultures in addition to mouse models. And nowadays also the engineered environments and microfluidic devices in 2D and 3D. And so we do multi-photon confocal epifluorescent microscopy, all of them in time lapses. We do lots of machine learning for phenotypic classification, and then distill this data into OD, PD models. Nowadays, we're also working on cellular POTS models. I got lucky and I fell in love with a computational modeler, so that can be kind of helpful. So we're practically looking in the tumor metastasis by in vivo, in vitro, and the computational techniques. So what's the simplest way that we can look into the tumor cell motility and the metastatic behaviors? That would be in vitro, and all of us know how to put the cells on the dish and look at them through time. One of the protrusion types that you can see in the migration of just cell on a dish would be lamellipodia. It's a part of the lateral migration. And then the other one is if you put cells on top of the thin or thick extracellular matrix, the structure is invadopodia, and it's been hypothesized is essential for metastasis since its discovery in 1998 by Suzette Miller, who I was lucky to have met her back in Georgetown, and she was one of my informal mentors and the person who introduced me into invadopodia field. So invadopodia top view would look something like this. In this picture, we have a cancer cell labeled by cortactin GFP, so one of the actin-associated proteins. And you can see a little bit of cortactin on the lamellipodia, on the edges of the cells, but it is mainly concentrated in this puncta that we call invadopodia. And then the cell is on top of the fluorescent gelatin, and you'll see in the later slide as well how these invadopodia form holes in the gelatin by degradation. So if you want to imagine how it would look like from the side, it's like a finger-like protrusion, and it's approximately 1 micrometer in width. So again, that is a really good challenge for microscopists because 1 micron is only a couple of resolution, a couple of pixels worth in the normal diffraction limited microscopy. So one of my questions became, what is the role of invadopodia in metastasis, and can we see that in vivo? And just to give a little introduction about how invadopodia lifetime and assembly actually looks like, precursor components come together, and they're actin-associated proteins, and ARP23 as a branching protein, and then TKS5, which is a very unique protein to invadopodia, and it's Sark substrate, and it's used for scaffold. It's a scaffolding protein. So all of these proteins come together in the precursor, and then the structure adheres to the extracellular matrix via adhesions and polymerizes actin in order to elongate. The degradation is initiated by calcium entry, which facilitates delivery of MT1-MMP to the membrane. And then we have the cycles of actin polymerization and depolymerization, which cause digging oscillations, as well as MT1-MMP degradation, which causes these holes in the extracellular matrix. So this process goes on for hours in vitro, and sometimes even days in vivo, as we found out. And so it's a pretty long-living structure. So that kind of gave us the hope that we can image it in vivo, because in vivo you cannot image too fast. And the approach we took is the multi-photon microscopy. And nowadays we had to actually build this microscope. But nowadays you can commercially buy them, either using one or two lasers. You can extend the spectra of the multi-photon imaging into 5 colors and the far red wavelengths and so on. So one of the tricks that we use that Jackie actually mentioned is photoconvertible fluorescent proteins. And we use them in addition to the imaging window that we first constructed with Jakob van Reenen when we were both postdocs at Einstein, and then he continued changing it and applying to all the different abdominal organs, and now actually Condilis Lab applied it also to the lung. And all of these windows can be used longitudinally, and as we've seen in a couple of previous sessions, nowadays we have a flexible PDMS windows in the field as well, which are potentially even superior to the ones we first made. So the photoconvertible protein can be photoconverted through the window, which is very convenient. We use Dendra simply because we had Vlad Verkusha, who's a person who developed this protein from the corals, the ocean corals. And this protein has originally GFP-like fluorophore, and then when you hit it with a 405 laser, it converts permanently to the red state. So you can follow where the cells came from and where they're going. So keep in mind also, like, this means that you need to have both confocal, so single photon and the 2-photon excitation on the same microscope. The 2-photon excitation of photoconvertible proteins doesn't work that well. So with this photoconversion, we could become a little more quantitative in terms of migration, intravasation, and proliferation. And here I'm just showing you first the static image of the primary tumor, primary breast carcinoma. And here the blood vessels are not labeled, so they're black, and the purple is for collagen fibers, and the green is for photoconvertible proteins. Now, if we go and start photoconverting approximately 100 cell large regions, one of them you can see here is right next to a major blood vessel, and the other one as well. Now, this one is very far away from the blood vessel. So this whole size is about 1 millimeter in reality. So it's 2 by 2 fields of view. And so if you come back the next day and image the same exact areas to compare what happened, you can see that these regions next to the blood vessel have actually aligned along the blood vessel, and some of them seem to have disappeared. So although we couldn't count every single cell due to crowding here, it seemed like some of the cells have gone, and they were not gone just from our field of view or from our stat, but they literally disappeared. So it was either apoptosis or intravasation, and as you'll see, it was actually intravasation that made them disappear from the field. In this other region that was far away from the major blood vessel, the only thing that happened was a slight dispersion. So yeah, they translocated a little bit, but they didn't leave the field much. So these were kind of 2 extreme cases, but of course, like, we've seen anything in between as well. And we wondered if some of these cells actually made it to the lung. So this is an image taken now 5 days after photoconversion, because we wanted to give them time to also extravasate and grow inside the lung. So this is a micromet of about 30 cells. And you can see some cells are still red, but some of them obviously have divided and they ended up being orange. So first time cell divides, it's going to have only 1/2 of the red fluorescent protein, and pretty much the same amount of the green protein, because the green protein keeps being made. So you end up with orange. The next time cell divides, you're going to end up with yellow. And if it divides again, it's going to end up being green. The green cells, however, it's very hard to distinguish between non-photoconverted or those that have divided several times. But we knew that there are micromets of the cells that we photoconverted, so we felt really convinced that we identified perivascular niche as a point of entry of the tumor cells. And we could do that in a pretty quantitative fashion. So another thing that we wanted to do is, of course, image these cells dynamically. And here I'm showing you the collagen fibers and then the non-photoconverted tumor cells. The dye has been injected here in red, and there's perivascular macrophages static all around the vessel. Now, if you run this movie for about an hour and then summarize the movement, you can see some cells have moved even more than 1 cell diameter or cell length. And we calculated the speed at about 3 microns per minute. So some cells are really fast. In the other regions, some cells are not moving at all, although the components are the same and the vessels are moving. Flowing. And this is where we thought, okay, this could be a candidate for invadopodia, and we started imaging 1 by 1 cell. And we've seen things like this. So little protrusions that are wrapping around the collagen fiber. And so this is kind of similar to what Giorgio was just showing. So they are actually even oscillating, polymerizing and depolymerizing actin in the vicinity of the blood vessel. Some of them are even pushing into the blood vessel. Now, if you look at the translocation, it's not there after 30 minutes or an hour. And it turns out that this movement is approximately 10 times slower than the contact guidance along the collagen fibers. So it seems like we have two different phenotypes, invadopodia phenotype being MMP dependent. So we could inhibit it with the MMP inhibitors. And in terms of the direction, it was always pointing to the blood vessel and the collagen fibers that are mainly aligned around the vessel, while contact guidance cared only about following the collagen fibers. Importantly, these two phenotypes were located in distinct tumor regions, so they practically were never present in the same field of view, which means tumor microenvironment is driving this. So we looked into as many parameters as we could of the tumor microenvironment to figure out what's driving them and start doing the correlations like PCA analysis, causality analysis, tree analysis, and nothing worked until we started collaborating with systems biologists that showed us that nonlinear classification by machine learning can actually distinguish between the contact-guided cells and the invadopodia cells. And if you look at this 3D plot, you can see that collagen fibers, so number of visible collagen fibers is one of the parameters. So it seems like, at least on this 3D plot, that if you start changing the number of visible fibers in second harmonic, you can actually change the behavior. That's like a child's view of the microenvironment. And so we started doing that, and it actually worked. So this is the control. So over 2-3 days, you can see same exact fibers. So the turnover of fibers in tumors, it takes about a week or two. But now if you start injecting them with a LOX inhibitor to stop the cross-linking, you completely lose invadopodia in the same exact regions, and you start gaining contact guidance. And we did the opposite with the glucose to increase cross-linking, and we got the opposite phenotype. So the switch between contact guidance and invadopodia is absolutely driven by the cross-linking. Now, what are the functions of these two phenotypes? Now we could actually separate them. So the contact guidance leads the cells to disperse, and of course they're proliferating as well. So that would be the search mode. And the invadopodia phenotype is leading cells to enter into the blood vessels. So that would be the destroy, basement membrane destroy mode, and intravasating mode. So invadopodia is the agent of intravasation, and its inhibition stops metastasis. It doesn't matter how you stop them, by MMP inhibitors, indirectly by cross-linking, or directly by molecularly abolishing the TKS5. And the take-home is invadopodia in vivo forms in perivascular niches when the tumor cell glides over collagen fibers and enters perivascular niche where the cross-linking is really high. It's going to start forming invadopodia in order to make the pores bigger and enter the blood vessel. And then it can switch back. Invadopodia is required for cancer cell intravasation, and its phenotype can be switched to contact guidance by cross-linking modulation. So we then got really interested in invadopodia and how it relates, because we all know that cancer is a disease of cells that are proliferating nonstop. This is where we got really interested in the cell cycle and proliferation. And I'm briefly gonna show you how the contact guidance in invadopodia are in the context of the cell cycle. So we use the Fuji probes for this, and we labeled our favorite breast cancer cell lines. And so with the Fuji, G1 is red, early S is yellow, and then G2 is green. Mitosis, that's about 25 minutes, is not labeled. So we placed the cells in the microchannels, PDMS microchannels. And we've noticed that as the cells are moving through these long channels, they go from red to yellow and green, and they seem to have changed their speeds. So we started doing the races of the cells, and we've seen that the red cells are approximately twice the speed compared to the green cells. So they're much faster. And how about invadopodia? Well, it seems that invadopodia is only really forming in the cells that are G1. Some cells that are S and G2, they do have precursors, what you call them, but mature degrading invadopodia simply doesn't happen in the other cell cycle phases. And you can see here a direct proof of that. So this is fluorescent gelatin in green. And you can see these black holes slowly forming, and this will be the quantification. So the cell goes from red phase, G1, into S and G2. And you can see how degradation, which is y-axis, happens only in G1. And precursors are here, these enrichments of the actin. So this was really convincing data, and we started getting into it a little deeper, but in addition to mechanism, we also, of course, wanted to show that this is true both in 3D and in vivo. We made the Fuji spheroids, and we showed that invasive strands are indeed initiated always with the red cells. Then the green cells start following. And in the later stages of the spheroid invasion, so this is a spheroid that's like 3-4 days old, you can see invasive strands have grown a lot. When you zoom into the tip of the strand, the red cell is still leading. Now, that was the case in 80% of the spheroids, but not 100%. And we were really annoyed by that. And we started now like looking into every single cell through time and when and why this is the case only in 80% of the spheroids. And what we've seen is that the red cells that lead, they have to divide as well. So every time that they're going through the cell cycle, they go back into the core, and the new leader comes about, and the new leader is red. So practically those 20% of spheroids that were in S or G2 were the cells in the period of switching. And I'm playing the movie here again. So red becomes yellow, and it goes back. The new leader comes. Becomes yellow, goes back, and so on. And you can see it here better in this graph. So red becomes yellow. When it's green, goes back. Second leader, third leader, fourth leader. So leaders switch position to maintain the cell cycle. This is something that we're working on modeling right now, but the cell cycle distribution and invadopodia is something that we then pursued also in vivo. And we've shown that these enrichments of actin are always in the G1 cells. And here, this is the 3D reconstruction of the perivascular niche, and I'm going to tell you what the colors are here. So the purple is the blood vessel. the enrichments of actin are invadopodia, they're little puncta, and you can see all these cells are red. So all the cells surrounding the vessel that are creating invadopodia precursors, and here on the right you have yellow collagen degradation antibody. So all the degrading cells are in G1 in vivo. And we understood so far just a little bit of the mechanism, and it seems that P27, which is the G1 controller, speeds up the turnover of invadopodia. So their lifetime becomes much shorter. So that means that they can go through these cycles of creating invadopodia, then moving further, creating invadopodia, moving further. So you have many more invadopodia because this turnover is much faster with the P27 localizing to the invadopodia. So if we overexpress the P27, the number of invadopodia increases a lot, but the average size decreases. And it's the other way around with the knockdown. So importantly, the palbociclib, which is the inhibitor, it's like called the magic drug, CDK4/6 inhibitor of G1, causes bursts of the invadopodia formation and the degradation of VCM. And this is something that we're looking into in terms of the translational application of the project, because palbo is used a lot so far in ER/PR positive cancers, but there are clinical trials done on triple negative, which is the type shown here. And we want to urge people, clinicians, not to use it in the patients that have invadopodia, because it would cause burst of metastasis. So this is the little overview. Since I'm running a little late, I'm not going to go into the details of the signaling cycle between invadopodia and the G1. I want to show you just a snippet of the paper that we just posted on bioRxiv. And it's about invadopodia in the collectively migrating and invading cells. So some of you may know that the epithelial cells go through several intermediate states in order to become mesenchymal in the EMT process. And so they can be stably EM, hybrid EM cells. Some people call it partial EMT. And 41 memory carcinoma model is one of such cells. It's a hybrid EM. So it has E-cadherin, but it also has Vimentin. It also has TKS5, which is expressed in invadopodia, and you can see the invadopodia here, and these cells grow as clusters, so it's very hard to separate them. When you form a spheroid made of 41, it has E-cadherin, and it's invading pretty quickly, and we're contrasting it here with a different clone that was isolated from the same exact tumor. So these are isogenic cell lines. The other one is called 67NR, and it's officially mesenchymal cell, but it doesn't have invadopodia and it doesn't invade. We thought that was quite interesting. And here I'm just showing you that the leader cells in the spheroid are actually the ones degrading and making TKS5 in the 41 cells. So what happens when you mix these two? This would model the heterogeneity of the cancers. Interestingly enough, they are able to cooperatively invade, but prior to cooperatively invading, here we labeled 41s with purple and 67NR with the green. And this is followed over 5 days. So the purple invasive 41 cells actually first sort to the surface of the spheroid, and then they start invading, and the green ones, non-invasive, are following them. Now, this is dependent on the cell ECM interface. So it doesn't happen in agarose. It only happens in adhesive ECM interfaces, such as collagen. And this is very similar to some of the developmental biology studies, but it hasn't been shown previously in the cancer cells, the sorting. In this cooperative invasion, again, 41s are always the leaders, and the 67NR are followers. Now, the most important question for us was, what happens if you inject these cells as a mix? So basically, can they cooperatively metastasize? And the answer is 67NR cannot cooperatively metastasize, but the 41 TKS5 knockdown can. And our hypothesis, and this is something that we're working on right now, is that 41 TKS5 knockdown, they're collectively invading and they have cell-cell contacts based on e-cadherin, so they can allow the cells to enter as a cluster, to intravasate as a cluster, where 67NR, because they're singletons, they cannot follow the 41s into the blood vessel. So they can follow them into the tunnels of collagen, but not into the blood vessel. And I hope I demonstrated that our approach can yield some interesting data in the cancer cell metastasis. I just wanted to show you that our data is now worldwide as BioArt, and I'm sure many of you have similar things. So they're all over the calendars and the galleries, and we're pretty proud of the, of this outreach of microscopy into the general population. And we had some exhibits in the museums and so on. And I would encourage all of you to do the same and to introduce the general public into the microscope, the beauty and the importance of the microscopy. Couple of people whose data was shown in this presentation have left the lab, and Lucien Perrin actually just started in Daniela's lab. And I would like to thank our collaborators and the funders, and we have open positions in case any of you are interested. And sorry for running a little late.
Thank you, Bojana. That was a nice talk. I'll be a little bit late. So there are a few questions. I'm not sure which one was the first. I actually think it was Guillaume. Guillaume Jacquin would like to know if your assay, if you tested whether there's a correlation between laser wavelengths intensity and the behavior of the cells in the intravital imaging.
Oh, he must be thinking of photoconversion.
Yes,
so yeah, we did extensive studies on this actually. Because we were trying to use multi-photon, and so we have a chapter that cites all the different powers and how you can photoconvert the cells. So mainly what it influences is the depth of the tumor that you can go to. Of course, we didn't go into laser ablation, but the higher the power, then you can convert one cell depth, 2 cell depths, up to 5.
Okay. Then there are 2, I think, quick questions and one more, maybe more important. One from Anna Campalans. Sorry, it has been shown that the G1 cells have higher mitochondrial activity. So do you think, or is it known whether mitochondrial activity is related to invasiveness?
Yeah, this is literally one of the experiments that we're working on right now. It's a great comment.
Okay. And then Takashi Nishina would like to know, but I think you partly answered this question. I think there are two types of breast cancer, benign and malignant, something that you might have addressed with the 61 NR cells. I would like to know whether there are any difference in the ability of invadopodia formation in these tumors. Are there any specific gene mutations for getting the ability of making invadopodia?
Yeah, we're not aware of any specific gene mutations that would allow emergence of invadopodia. And so far, people have been thinking about invadopodia as something that only happens in mesenchymal cells, because it's actually a good marker of invasiveness of the cells. And so this is the first time that we're, now realizing that, the partial EMT cells can also have invadopodia. So obviously they have to express TKS5, and they have to express at some higher level MT1 MMP, and then MMP2, which gets activated by MT1 MMP. But yeah, I mean, SARC is one of the drivers. Honestly, invadopodia has like over 60 proteins involved. But yeah, malignant cells have invadopodia, benign don't. So also in terms of tumor cell types, ER/PR positive cells don't have invadopodia, but we know of examples of HER2 positive cells having invadopodia, and also triple negative.
Then the last question from Roberto, which is also something you partly answered. Any difference in composition between in vitro invadopodia and 3D invadopodia? I think it's still a major question, I think, in the field, right? Whether there's also a good marker for detecting invadopodia in vivo.
Well, TKS5 and the degradation would be fairly good markers. We know that, like, actually Daniela's lab is maybe, one of the only studies that showed artificially like growing invadopodia up to 10 micrometers in vitro. So you could see where the different invadopodia components are, like cortactin versus actin versus MT1 MMP, where exactly in the length they're going to show up. But not that we know of. The composition is different. Like, it's only the appearance actually that it's different in the in vitro versus in vivo.
So I think we'll have a shorter roundtable today because we're a little bit late. I think there was one remaining question from for Franck.
Yes, there is. Takashi Nishine has two questions for Franck. It's like, you're saying why blood vessels do not infiltrate tumors. I think you showed that while they grow, tumor vessels are not, mass is not increasing. Is this particular to glioblastoma or some other tumors?
Yeah, I think that's not exactly what I said. I pointed to the fact that there was an existing network of blood vessels, and despite the fact that there was an increased proliferation of cells, this vessel structure did not develop further, and most likely it was squeezed. At least that's the way we interpret the data. The density of cell was such that the vessel would have been squeezed and with very little blood flow inside. So that's more the message I wanted to carry.
He's also asking, do you know the source of VEGF in your model?
So the source, the tumor cells by themselves already deliver VEGF. Macrophages are known to release VEGF, and possibly endothelial cells as well. So I mean, I think it's a soup, and probably the different cell partners contribute to this production.
Okay, these are the last questions that we had. Maybe Jackie, I knew that you wanted to launch something about the number of cells. right? What's imaging?
No, yeah, there were a few numerous cells. No, there were a few things that were maybe like I would like you guys to discuss since you are experts in particular field. Maybe one aspect for which I would like to have your opinion also is what is the best method? Because I think Franck, for example, started his talk by mentioning that is how to be able to do longitudinal imaging. You mentioned vasculature as anatomical landmarks. Is this something that is reliable, especially in the context of vascular remodeling? What are the other options that you might have?
So just to come back on the vascular landmarks, these were taken outside far in the field of view, far from the tumor, and in any case, the tumor size is known, and all the border are with cement. So in any case, you are always able to come at a certain distance from the border. So at least geometrically, we can be exactly at the same position. And then the only thing we have to face is indeed the remodeling of the tumor eventually. I mean the squeezing. I mean what Giorgio was mentioning in his talk.
Do you want to comment further, Giorgio?
Yes, I mean now we are trying actually something new. We are trying to use the grid cover slip. It's something that is really recent for us, but in that case you can easily go back in the same position, and that helps. For sure we use a lot of the vasculature because we use the normal vasculature of the surrounding tissue that is not changed too much. But I agree, when the tumor grow a lot, I mean you cannot see anything anymore. The other thing that I wanted to just quickly add is about the other technique. There is no multi-photon, there is optical frequency dynamic imaging. In that case, you can see the whole kind of window. So in that case, I mean, you set just the kind of window, and so as a side. But in that case, for sure, help a lot the longitudinal imaging of the whole and different regions of a larger field of view.
Okay. And then add another subject of discussion, but you guys can comment anytime, is the photoconversion that Bojana, you showed today. What are the optimal tools that we have in hands right now? Because you mentioned, for example, the need for having a single photon excitation for the Delta-2. Are there additional options upcoming that makes life easier when we want to use photoconversion in any type of intravital imaging experiments?
So in our case, we have a hybrid confocal multi-photon microscope, so that makes things really easy. But another way to go is just to have an LED that you can use for 4 or 5 photoconversion. Now, there are, of course, other proteins. I was just showing the green to red photoconversion, but there is the photoactivatable GFP. I mean, that one for me, it doesn't seem like a convenient one because you go from darkness to GFP, and then you have the photo switchable CFP GFP. Which for those who can separate CFP and GFP is actually quite convenient, and it's working really well. And there's another one that's photo switchable orange to far red. So that one you can photo convert with 488. So yeah, there are absolutely other options in you know, you could even combine 2 different cell types that are interacting, and in one you have CFP GFP, and in one you have orange and far red, so you can convert both of them. So I'm just going to add, like, for us, the vasculature was never a great landmark because the breast carcinoma grow like crazy, and from one day to the next day, vessels change so much that there really is no way to rely on them. So, I mean, in our case, we just use mostly photoconversion. So we know exactly how because we make an array of regions, sometimes 4, sometimes 9 or 12 of them that we photoconvert under the window. And then we know exactly how they're located. And the tissue also stretches and changes shape. So that's why it's you want these landmarks to be sort of relative. The only other way that I've been doing is the laser ablation, which is like a tattooing of the tissue with really high power laser, and then you make 4 corners. And even if those 4 corners are not square anymore, the next day they're kind of twisted. You know relatively what your previous coordinates are.
Okay, cool. So, and then I have one last point that I'd like to raise and get your comments of all of you, the 3 of you, is, and it was raised by Roberto today as well, is the potential use of genetically engineered models, right? To, I guess, better mimic slow growth of tumors. So what's your take on that, and how difficult would that be? Have you guys ever tried this type of imaging on this type of models?
Yeah, in glioblastoma is very difficult because there is not a very good model for genetically. It's not a model that we call everything that is happening in human. It is true for probably every type of tumor. But in glioblastoma is really, really difficult. There are models, but it's not. It's definitely not something ultimate at all. But it's definitely something that is needed because the microenvironment changes a lot when you inject a cell. Massively you induce inflammation and everything. And so, yeah, I agree.
And that's the idea. You move away from this bulk injection of a huge amount of tumor cells that might drastically modify the microenvironment.
Actually, linked to that question, I had a question about glioblastoma, maybe a naive question. Does this have a particular preference to grow in the brain? Is there, you're always talking about niche, but does it grow anywhere?
Okay. Where you inject, if there is a, yeah, that is also the other problem, is a big problem in intravital microscopy in glioblastoma. When we inject and we want to follow, we need to inject in the cortex because otherwise, if you go deeper, you don't see by intravital. But in general, the good model of glioblastoma are never in the cortex because glioblastoma not very usually grow in the cortex, and so is something that you cannot get rid of for any other choice. But yeah, that is a big pitfall in glioblastoma field in intravital.
I think it's touching a little bit of that is always a model, right? But
it's always a model. Yeah. Indeed, the other thing that I personally always combine, but I guess all of us is doing with other models, with histology, with everything that you do. Not, you always need to have another model that confirms your intravital microscopy. There is observation.
But why do you think it doesn't happen in the cortex? I think that's a problem.
Well, we don't know. We have no idea actually. But in general, it happens also sometimes in the cortex, but it's not usual.
I think in this concept of vessel cooption, I'm really wondering whether the cooption that is existing in the context of glioblastoma is similar mechanistically and molecularly speaking to cooption that happens to brain metastasis. So do you guys have any idea on that?
I can tell you that vessel cooption happen a lot in simplifying, happen a lot in tissue that are highly vascular, and so has been shown in lung, has been shown in liver and in brain. So for sure, having a lot of blood vessels make obviously help in that. But I can tell you that there are other papers that will show very interestingly brain metastasis, and so the mechanism in the brain are very similar. Okay, and so we also see L1CAM as an important that was shown in breast metastasis in the brain, also important in glioblastoma. So really, the environment, the organ, is doing a lot. It's really doing a lot.
Actually, I had a question for Franck because he was talking about the immune switch. Is this related to tumor cell death, permeability of the vessels, so permissiveness? Do you see leakage in that sense? Why the switch, and what can you?
We didn't see a specific leakage, at least with the rhodamine dextran that we are using. That is pretty high, and that is 70 kD. So at least with this big molecule, there was no obvious difference. But we didn't study carefully this at the moment. Yeah, I mean with different molecular weight to really be sure that this would be correlated or not.
And Franck, in these experiments where you perturb the immune switch, so you address the chronology of immune cell recruitment, you perturb it with bevacizumab at some point. Have you done similar experiments where you do an anti-stromal treatment, as you've shown in the beginning of the? No,
we didn't do yet. No, because the anti-stromal is more difficult to use. I mean, it's twice a day you have to give the injection for a very long time. So whereas the bevacizumab can be used more easily. So at the moment, no, we didn't do.
I also do a question to Franck. You show everything on GL261, right?
U87 and
no. Yeah, okay. for the immune part.
Yes.
Did you have the chance also to try with other model like CT2A that have different immune components? Maybe it could be also interesting.
We didn't do so far. Indeed, I mean, by this model, we had the feeling that the node to be addressed would be the dendritic cell, the monocyte-derived dendritic cells, and I mean the research at the moment is more to target this, but even in some other model of neuroinflammation, that would be more simpler to address. But we would go back to glioblastoma and difficult model afterwards. Yes.